A little bit about Keith. Keith is an experimental and computational neuroscientist at Washington University. He received his PhD at University of Wisconsin-Madison, and he worked with Dr. Gina Turgiano.
That's close enough.
Close enough. At Brandeis University. Keith joined the department of biology at WashU in 2017. Has been working on many number of interesting things, such as the kind of neurocomputational underpinnings of Alzheimer's, nature of sleep, which we'll talk about here. I personally had the pleasure of working with Keith as an LEC mentee, where we've studied behavior and cognition in warm-blooded mice that have learned to hunt cold-blooded insects over a thermal gradient. Just as an aside, kind of one of the cool, slightly sci-fi things I've seen in Keith's lab. His students have performed surgeries where they remove like the skull of a mouse and attach electrodes to the mouse's brain. Yeah, kind of really cool neural recordings. It just feels like science fiction to me, but I guess it's normal for them. And as an ecologist who does a lot with like diversity, species, and actions, I'm really excited to learn something pretty fundamental to animal behavior, which is the nature of sleep. So with that, I'll let Keith give his talk. A waste of time or a chance to reset the OS. Why do animals sleep?
It's got a red light. Oh, there we go. Can everybody hear me? Okay. Thank you very much for coming out here on such a beautiful afternoon/evening. My name is Keith Hengen. I'm an assistant professor of neuroscience in the Department of Biology here. And my lab is sort of generally focused on the mathematical principles that give rise to complex biological computation. And we're sort of specifically interested in how those processes become robust across very, very long periods of time, specifically decades or your lifetime agreement. And so I'm just going to go on a limb for a moment and assume that many of you are wondering why I'm, given the last 20 seconds of my monologue, you might be thinking, why are you speaking at the Living Earth Collaborative? And normally I would be wondering the same thing. But for a happenstance collaboration involving Tony Dell over at NGRAC and John Grady, now a prestigious, esteemed LEC postdoctoral fellow, and because of those two, and my graduate student, Jacob Ahmed, is Jacob here? He's a class. Where is Jacob? Okay, Jacob should be here. Because of them, and I am now one of you. I'm a member of the LEC, as odd a bedfellow as I may be. I'm in your family. So with that, let me just dive straight into this cool project. So, oh, somebody clicked something else. There we go. Okay, so this is a mouse. And overlaid on the mouse are some circular dots, and that's the result of some convolutional neural networks and like deep vision, whatever algorithms that track points on the animal's body. And that's not relevant. The thing that you need to know is that this mouse is famished. She is very, very hungry because we have not let her eat food for the last, 14 to 16 hours. She's been food deprived overnight. So she's cruising around, waiting for lunch to show up. And fortunately for her, we're going to deliver lunch because in drops a Tajikistani red runner cockroach. And so this mouse identifies the cockroach, says that looks delicious. She pursues this thing. I think right about now she rips off a few of the legs. She'll turn around in a moment and go back and consume this. And so from my perspective, this is phenomenally interesting as an act of sort of spontaneous, incredibly complex biological computation. This mouse has been able to sense its own internal milieu. It's hungry, attach that to a set of potential behavioral outcomes, and then say, well, I'm not going to start eating the corn cob bedding. I'm not going to eat that metal pipe that sticks in here. She waits for the cockroach to get in, and then she has complex object recognition. She maps out a set of ways to get to the mouse in space. She pursues it. She doesn't tip over. She doesn't have a seizure. She does all of this every single time, flawlessly. She never mistakes a thing and starts attacking the wall. This works robustly. Okay, this is amazing. John thinks it's cool for a totally different set of reasons. John says, damn, that's an endotherm and an ectotherm. And I'm not sure which is which, but one is cold-blooded and one of them is warm-blooded. And I guess that warm-blooded and cold-blooded species interactions, trophic interactions for the nerds, have a lot to do with species diversity, biodiversity, and speciation across the planet. And it's very, very hard to get at these types of interactions. So metabolic theory of ecology, yeah, okay, not astronomy. That's a joke. Metabolic theory of ecology basically is restricted to studying cellular processes, muscle contraction limits, things like that. To actually then say, how do metabolic differences across the temperature gradient factor into complex trophic interactions between warm-blooded animals and cold-blooded animals? It's a lot harder to achieve. So we joined forces to do this, right? And my understanding is, can animals actually take that information to make predictive decisions about upcoming interactions? So for example, would a mouse be more likely to hunt in an open field on a cloudy day than a sunny day? Because on the sunny day, the cockroach is going to be hotter and moving faster. The odds shift in its favor, right? Turns out that's exactly what happens. But we run to the crux of the issue, which is that John doesn't want ecology talk today. Right? So I called John in my office and I said, John, I've never talked about ecology. I've never spoken to ecologists, and I've never shared any of these data. What should I do? And he's like, you're in luck. I don't want you talking about that. So we're gonna take a hard left turn. And this is the switcheroo, right? The bait and switch. I'm sorry. So now the alternative title of my talk is Hardcore Systems in Computational Neuroscience for the Curious Ecologist. So, ready? Let's change directions. So with that, one of the central questions that my lab has decided to focus on in the last 6 years now, oh my God, we need to publish, is what I would assert is one of the kind of grand remaining questions, at least in neuroscience, but I would argue in all, at least animal biology, right? So this is, I think this is a big deal, like capital B, D, big deal. And that question is, why do we sleep? Right? We know we need to sleep. If you don't sleep, you will die. Have you ever heard of fatal familial insomnia? Somebody heard of that? This is awful heritable genetic conditions of family in Italy that gets it. Starting in their 30s, they start having trouble sleeping, and it gets progressively worse and worse until they die. It's terrible, right? You need sleep. We don't know why. So since this has very little to do with ecology, here's my brief outline. So one, we're going to have to cover a little background on the sort of state of the art, the cutting edge of the field of sleep, so I can catch you guys up to speed. Fair? Okay. That's sort of a predicate for where we're going to go. So buckle up. Two, we're going to have to go over some first principles reasoning of why my team thinks that the state of the field of sleep is bullshit, for lack of a better term. Three, we're going to have to then go over a quick introduction to emergent dynamics, because I think that this is where the solution to our problem lies. Okay, and then I'm going to get into the data to try to kind of close the case on this thing. So if it feels like we're going in some very, very non-ecology directions, that's because we are. And I apologize in advance. Okay. But why trust me when I say that we don't know what sleep is doing? I think the authority on this is a guy named Bill Dement, who died recently. So the late, great Bill Dement. So Bill Dement is an authority because he discovered REM sleep. So do you guys know what REM sleep is? REM sleep is when it's rapid eye movement sleep. So when somebody dreams, if you are creepy and you're watching somebody sleep, you'll notice that they go into a phase of sleep where their eyes start moving all over the place, right? It's a very physiologically distinct state in the brain. It's kind of conserved across birds and mammals, and it's called REM sleep, and it was discovered by Bill. Bill also discovered and named the 5 stages of human sleep. He founded the American Academy of Sleep Medicine. And most importantly, he had a walk-on cameo in, here's your pop culture quiz, Mike Birbiglia's Netflix direct movie, Sleepwalk with Me. I'm not gonna, I got one nod. Yeah, Mike Birbiglia is funny. He's a good comedian. I recommend him. So nonetheless, those are some serious credentials. So obviously before he died, so recently, but prior to death, Bill was asked, why do we sleep? This long career studying this, founding all these things, all this attention and everything. Why? Why do we sleep? And his answer is spot on. He said the function of sleep is to reduce sleepiness. And I think in this snarky quip, he nails it. Because I think he's basically saying something happens to us. It's very, very compelling. And we need to sleep, so we do. But it's just to deal with whatever that thing was that we call sleepiness. And that's it. So after I would argue 100 years of investigation, we have no idea. But as long as there have been people, there has been sleep. And so people have wondered why we sleep. And we still don't know fundamentally at its kernel what's happening. So I can formalize this question a little bit more formally for you. And that is to say, the search for a core function of sleep. And this is important. that which explains the restoration of neural computational capacity, brain function, has been called, in very kind of theatrical terms, a worthwhile but elusive phoenix. Okay, yeah, so Giulio Tononi and Chiara Chiarelli are sort of leaders in this field at the University of Wisconsin-Madison. I've rotated with them as a grad student. I'm like simultaneously friends with them and terrified of them, but they're the Italians, is what we call them in the field. That's the Italians. It's them. So they point to this as being a thing that needs to be pursued, but like consciousness has yet to be solved. Okay, and the best thing we've got is well, okay. Sorry, this is why I wanted to presenter mode. So what do I say? First, I'm tell you what we do know, and then the best model that we have. So what we know is that every single thing changes, and it's very, very hard then to make insightful progress into a complex system when every aspect of that system changes. So every time you fall asleep and wake up, everything changes. Genes change, proteins change, cells change. So as an example, here's a paper showing that proteins in synaptic membranes are changing systematically with sleep and wake. This is showing that the electrical currents passing through neurons are changing as a function of sleep and wake. This shows that the actual cell morphology, how neurons connect to each other in the brain, changes as a function of sleep and wake. So everything is changing. One of the big systems that's gotten a lot of attention at WashU, and especially over the last 10 years, is something called the lymphatic system. So it turns out when you fall asleep, the extracellular space in your brain expands. And it's like flushing a giant neural toilet. All of the kind of accumulated junk, so metabolite, byproducts, misfolded proteins, flush out during sleep, right? So all of these things are certainly happening. I'm not debating. I'm not suggesting that there haven't been many, many, many processes that have co-evolved to take advantage of the sleeping state. But the issue is that none of these things intrinsically explains why you feel like $1 million after that 8 hours of uninterrupted, glorious sleep that I never get anymore, right? None of those things, you can kind of make it a 3 or 4 degrees of connection, maybe this leads to that, to that, to that, and it's kind of this hand wave thing. So none of those things directly explains this. And so then we're left back with what Bill Dement says, that the function of sleep, a restorative function of sleep is unknown, and it might as well just be to reduce sleepiness. Okay, so this is the best that we've got. And that is a 41, is it 2023? Yeah, 41-year-old model that's purely heuristic. So this guy, Alexander Borbely, came up with this in 1982, and he said that sleep is determined by 2 processes, process C and process S. Process C is the circadian process. So has anybody ever flown to Europe? One of you, wow, real world travelers in the LEC. Okay, well, did it suck when you first got there?
Yeah.
Why?
Jet lag.
Yeah, right. So you have an internal circadian rhythm that determines when your body wants to sleep and wake. And when it is misaligned with the external world, it is terrible, and we call that jet lag. Right. So at least one of you knows that that process C is very, very distinct and very powerful. So the other process, process S, is the sleep homeostat. Welcome. I'm glad that we filled this up. I'll just give you guys a warning. This has nothing to do with ecology. Have fun. Okay, so process S is referred to as the sleep homeostat. And again, this is a heuristic thing. We have no idea what it is, but at least it's a formal term to describe something accumulating during waking and then decaying during sleep, or decaying during waking and then reaccumulating during sleep. And it is presumed to be the overlay of these 2 things that sets up why and when your brain needs sleep, right? So I was thinking, okay, I need to like dig into this for a bunch of ecologists, and I found a great website, mattressadvisor.com, so, to explain to you why you need to sleep and someone trying to talk a mattress to you. So this person, Lauren Bullock, nails it. Lauren says, the sleep-wake cycle is regulated by 2 separate biological mechanisms in the body. I think that's implied, but whatever, we'll give them a break, which interact together and balance each other. This model, Lauren credits Borbely. It's the citation, right? Alexander Borbely positive thing, and she then brings up the circadian rhythm known as process C. We just went over, and here's where things go off the rails as far as I'm concerned. Sleep-wake homeostasis, process S. This is spot on accurate for how the world thinks about this. Represents the accumulation of a hypnogenic substance in the brain, which generates homeostatic sleep. I call bullshit. I think that's insane. There's no way you can convince me that all animals across all phylogeny and all of life and all of experience and all brain regions and every possible combination of those things have a single molecule that is increasing every time they're awake, and we haven't found it yet. That's nuts. Okay, that's my aside on that. So, which leads us then to a first order reasoning process to say, if it's not that, and people look for this hard, there are countless papers saying, well, this molecule goes up, that molecule goes up, and then somebody else says, no, it doesn't, it goes down, right? So all of this disagreeing stuff, and nothing really makes a good impact on this. Nothing has explanatory power. It all feels kind of abstract and arbitrary. And so that leads me then to say, if we break down the problem, the first thing is that evolution can only select for behavior, right? So sleep seems to be an evolutionarily stable thing. All animals do it. Even worms sleep, as some people will assert, but everything sleeps. I don't know about plants, but worms on up. Rachel, you give me a no? Okay, we're gonna leave plants out of the picture, and I'm sorry if that offends half of the LEC, but everything else sleeps. So evolution can only select for behavior. So my argument is that sleep must subserve something about behavior. And it has to be something very, very deep. It can't be a specific behavior, right? So a lot of people have posited that perhaps sleep is involved in memory replay and memory consolidation, right? So you study for tests, you need some sleep so you can do well on your exam. And the problem is you don't study for a test every week. So by that logic, when you don't have a lot of memory to consolidate, you shouldn't need to sleep. You should have months of your life where you don't need to sleep, right? But that doesn't happen. So there's got to be something fundamental that is not tied to a specific behavior, whereby behavior is affected systematically by sleep and wake, right? And the problem here is that behavior arises not from a cell or a protein, right? You don't have that's my mom neuron, right? It's you guys have 86 billion neurons in your brain. And so to recognize your mother involves billions of neurons to go from transducing the signal of photons into your retina and then sort of forming a face and then getting the object recognition and who is that. So that's a very, very sort of complex set of computational processes that occur. And so the point that I want to make is that the thing that's going to, I think, the thing that will necessarily trigger the need for sleep has to arise at a systems level. And that might then be subserved by a more reductionist change, right? So the systems level effect in behavior could be subserved by plugging a protein in or out of the membrane, but the thing that's going to matter to the animal and to evolution is the system's dynamics. Does that roughly make sense? Getting a couple nods. Okay. So then I think that we can come into this and say, well, sleep must be a systems level solution to a systems level problem. And so perhaps by going deeper than that and getting too reductionist, but trying to restrict that to a protein or to a gene or to a microRNA, we're missing the boat entirely. So our hypothesis then is waking degrades the operating system. So whatever the kind of fabric is that gives rise to sensation, perception, cognition, and behavior, the kind of computational principles that allow all of those things to unfold in all conditions, all the time, flawlessly, that must somehow be corroded by experience and by waking, and then sleep must reverse those processes. Okay. And so rather than going on a fishing expedition, because I told you everything changes, I think this has to be approached hypothesis-driven first. And so we can take a control theory perspective to this problem. So control theory basically stabilizes airplane wings. It's a very sort of engineering thing. The idea that is central to control theory is that if you have a set point and the system deviates from that set point, it's going to generate, you need some error signal. You need to sense that error signal and then feed back onto that system. So think about your house. What's your house set at temperature-wise?
Like 71.
71 degrees. That sounds comfortable, right? That's a set point. So your thermostat, if you leave the windows open and it's last week, it gets very hot in your house, it's no longer 71. Your thermostat detects that. The error signal is the differential between 71 and the 9 million degrees that was St. Louis last week. And then that kicks on a set of mechanisms in your air conditioning or in the winter, the heating elements to restore that 71 degree set point, despite the fact that you've left your window still moving. Okay, so The key point here, though, is that we should be able to establish a set point and an error signal, and then make predictions about how the system should function if we've identified it does correctly. And ideally, that set point would even have explanatory power. It wouldn't just be arbitrary. Okay, so that I know that that was very vague. So we're gonna get a little more. We're gonna get into the weeds now. So on to the kind of emergent dynamics. So we're going at this thinking, okay, well, what might represent a set point that has true computational implications across the brain, across phylogeny, and across behavior? And so there's a set of features that we think a brain must, that evolution of a brain must attend to. So I'm trying to do this again for ecologists. I'm like trying to pull jargon out, and I'm stumbling. That's why. So number one is dynamic range. So dynamic range is the difference between the strongest signal and the weakest signal that a system can faithfully encode. So think about your visual system. If you're in a dark theater, you see fine. And you walk out into bright sunlight, you adjust for a moment, you have millions-fold difference in the numbers of photons hitting your retina, but you can accurately transduce visual stimuli across that. That is the dynamic range. So there's pressure on systems to have a broad dynamic range. Secondarily, you need to have some form of maximizing the communication of neurons over distance. It's not going to work. So your frontal cortex and your visual cortex are 10 centimeters apart. So you need something that's going to maximize the ability to propagate signals through those 86 billion neurons. You need some way to maintain signals over time, right? You need to be able to react to a quick noise, or you need to ruminate on a broken heart for weeks to months to forever. And then you also want to have the ability to represent lots of pieces of information. So you could imagine in a seizure, for example, if every neuron in your brain spikes at the same time, they're all redundant. You only need one neuron for that, right? So you basically have a two-sided die. The alternate side of that is you have 86 billion neurons, which means that at any moment in time, you have 2 to the 86 billion possible states that your brain could exist in, right? So that's a lot of different things that your brain could potentially encode. And we're not even bringing time into the equation here, right? So if you want to sort of simultaneously maximize those things, there's one answer to this question. So I went overnight, so I knocked on the doors of a lot of people in the math department. I talked to people in theoretical physics, and there's one answer. And this, so sorry, when we put these things together, we're going to call that sort of the computational capacity of this network. And so those things arise from a very specific regime of excitability called criticality. So again, I told you we're going to take this little trip. Stop me if you have questions. I doubt that I'm being as clear as you're telling me now, but this is best explained if we think about magnets. So you guys have seen a magnet before? Okay. So in a magnet at room temperature, all the iron molecules are what's a dipole, right? Dipoles, they have an orientation to them. So they all align together and they point in the same direction, right? So what that means is that the interaction magnitude between these is very, very strong. So if we were to take this one and flip it, the neighbors would grab it and pull it and flip it right back in that direction. So think about this from this perspective. Because the interaction magnitude is so strong here, if I tried to encode a signal at this arrow and you were listening here, flip it down, flip it up, flip it down, flip it up, are you ever going to see that? No, you can do it a million times. It's never getting through that network, right? The change is entirely localized. But if we heat up that magnet and we get this boiling hot system, it's no longer magnetic, right? Because the iron molecules become white noise. They don't align anymore. And at this point, if I flip this molecule, it has no impact in its neighbor. There's no interaction strength. And you just have this kind of white noise. So again, if I start flipping this molecule, you're never going to decode that signal over here. But interestingly, there is a very, very sort of bizarre and non-linear state that exists at the phase transition between ordered and disordered, right? And that is called criticality. And at criticality, you maximize all of those features that I just described to you in the last slide. So you maximize entropy, you maximize dynamic range, you maximize the ability to send information over distance and across time. So what happens in the iron molecule is that if you were to flip this, you see kind of a swirling pattern of rotating iron molecules traveling through the whole thing. It wouldn't cascade into disarray, it wouldn't become chaotic, but it wouldn't immediately close down that signal, right? So in the brain, you can think of the same thing as potentially occurring. But instead of hot and cold magnets, you can think about a phase transition between a heavily inhibited brain. So if you've ever had alcohol, you understand inhibition, and say a regime of excitation, something more epileptic or chaotic, right? And so exactly at that phase transition, you get the same thing. You get a maximization of dynamic range, of information transmission, information encoding, and you can actually do this in synthetic systems and artificial systems. You actually maximize the computational capacity to solve complex problems in artificial, like, neuromorphic, you guys don't know what neuromorphic chips are probably. All of the AI stuff, if you tune those underlying models to be a critical system, you will use electricity and solve problems faster and be less prone to things like catastrophic forgetting. So this is a fundamental computational principle that then exists in nature as well. And so we might make the hypothesis that this is the operating system of the brain. So I want to just give you a quick cartoon we made to show you what this looks like. So if the system is in that inhibited regime, and we drop a bunch of blue dots into this grid, the only rule I've set is the probability that a dot can propagate into the next square. And so what you see here is that no matter how many times we seed this model, these spikes, these dots, these, whatever you want to call them, they just sort of die out. They percolate a little bit and they die out. So again, if you were listening here, you're never going to hear it. You're never going to get that signal. Now, similarly, if we go to the other end of the spectrum, to the supercritical regime, you go to the supercritical regime, everything blows up, and again, you can't decode a signal. But at criticality, what we see is that the signal, the spikes, the blue, is sort of self-perpetuating. It never grows to consume the entire thing, and yet it never decays and falls silent. And so it's at this point that we think brains must be organized. So I'm going to skip over this and say we established. So in 2019, we had a paper that was, I don't know, one of the coolest things I've ever done, showing that not only do brains do that, but it's a homeostatic set point, kind of like your 71 degrees in your house. So if we actually push a brain out of that critical regime, it will actively push its way back there. So we thought, okay, this now seems like this might be the answer. So my graduate student, Ifan, did the sci-fi experiments and implanted, sorry, is this gross for ecologists?
No, not at all.
Okay, all right. I was wondering, do I need to kind of?
No.
Okay, we can magically detect neurons. No, so we put a whole ton of little microwires throughout this rat's primary visual cortex, and the rat then lives for a few months afterwards, and we can actually track the activity of individual neurons across many weeks of recording. So we can find every time a neuron fires a spike. And we take advantage of the fact that rodent sleep is highly fragmented. So you sleep, I don't know, 8, 7, 8, 9 hours a night, and you're awake the next day. You don't. I just saw that. But here's a map of one rat across. I'm showing you 5 days out of a 10-day recording, and so this is 12 hours of light, 12 hours of dark, and each circle on here represents its area represents the proportion of that hour that was spent asleep. And so what you can see is that rodents are generally considered to be nocturnal, and so there's an enrichment of sleep here, but it's certainly not what you would think of when you say it's a nocturnal animal, right? So the cool thing is we can actually take advantage of this intrinsic variability by just recording long enough that we can find every possible combination of time of day, amount of sleep, prior history, et cetera, et cetera. So we can take the animal's own variability as a set of experiments and internal controls. And so in this context, here's what the raw data looks like. You guys probably don't care quite as much, but this is a voltage trace, and each one of these little hairs is an action potential from a neuron. So for those of you who've never taken intraneuro, neurons fire an electric spark, and that's how they encode information. It's a 0 or 1 signal. And so we can take all of these and do a bunch of processing and math, and we can actually get the kind of fingerprint of each neuron, and we know when it was communicating with its neighbors. And so we're going to use those data then to examine this. So this is sleep. And so I look at that, it's like the matrix, and you see the zeros and ones go, that's sleep. So this slow down, up, down, up, down, up, that is the slow wave of slow wave sleep or non-REM sleep, right? And so in here, you can see that there's some organization of this activity. So we can take each of these things and we can basically make a raster plot. Let me start. I'm going to get a little too into the weeds on this. Okay, I'm trying to like read the room here. So the key thing is this. Let me just go back. In this situation, we have to click it. In this situation, the key prediction is that, let's say you were listening at a few of these squares. the sort of magical thing that falls out of that phase transition is that you get a power law. So if we count the number of times that blue is kind of crossing one of those points, usually it's just going to be 1 or 2 blue dots. Occasionally you might see 10 or 12. But if you listen long enough, you'll see 1,000, right? So you'll actually see a power law distribution whereby there is no spatial or temporal scale of these swirling patterns. Versus in this one, you're never going to see 10,000 go by your electrode. And in this one, you're never going to see 3 or 4. It's just going to be a big swirling mess the entire time. So that's the kind of fingerprint we're going to look for, right? And so if I sort of zoom ahead, I'm going to skip over the details because I don't think you care. You can ask. This is that power law. So when we look at those spikes moving past our electrodes, and we look at the probability versus the number of spikes, it's a power law, right? It's the definite. So power law. John, if I have a square and its side is 3, what's its area?
It's 3 squared.
Right, which is
9.
right. And if its side is 10,
It's 100.
it's 100, right? And what if its side is like 16 million? 16 million squared, right? So that's exactly that is a power law. So it is a power that relates to variables. And the cool thing about power law is that you don't have a different set of rules for very small squares than very, very large squares. Now, most things in biology are not scale invariant or time invariant, right? You will never see a heart rate of 4,000, at least in a human, right? You're never going to find a 4-inch tall person who's at least an adult. There is a typical spatial and temporal scale for many things. And so a critical system is noteworthy for the fact that it is scale free. So think about it this way. If these little spikes passing by our electrodes are kind of the letters with which the brain uses to write prose, words, sentences, paragraphs. In a subcritical system, you're restricted to very short words. And in a supercritical system, you can only use huge, long, rambling nonsense. But it's in that critical system where you're able to kind of use the entire dictionary. And the key thing is that we look for this. So we can predict, so just skip over some things for a moment. We can predict this orange line. So based on the power laws we measure, we predict this orange line, and then we project our data onto it. And we can say, how much do those two things deviate? So we came up with, this was our big hit in 2019. We came up with this deviation from criticality coefficient. So it's the first time we've ever been able to really quantify how close or far a system is to that critical point. And so what I'm trying to show you here is that this DCC value is pretty low. It's below 0.2. That seems to be our magic number. And doing that in both sleep and in wake. So if we look at this DCC value and we say, does it vary over time? It does. There's a lot of variability in here, but it doesn't really seem to map on to light and dark cycles very much whatsoever. It just seems chaotic and noisy, right? But if we zoom in on this, if we zoom in on 12 hours of this, here, so again, this is a rat running around, looking at electrodes in the brain, and we're basically saying how close does the brain look to criticality? If we, instead of breaking it down to this binning, we now look at this measurement of distance from criticality as a function of behavioral state. Here you can see that the system's getting further from criticality. It's a distance, it's like a golf score, right? Larger number further from. Does that make sense? Okay. Green is active waiting, so it's getting further away from criticality. Jumps up, seems to go up a little bit, jumps up a bunch. Sleep is blue and gold. So across this period, with as much as sleep as quiet awakening, it drops down. Here and here are lower. That's the drop down a bit, drops down a bit, drops down a bit. So behavior is very, very, very complex and highly variable. What you do at any moment is very high dimensional. And so despite that, it still seems like there's some trends there. So we go back to the control theory thing. If we're right, I would posit that the most stringent test of whether you've got the set point and the error signal correct is if you can predict the future behavior of the system. So we go back to your house and you open that window. If I know that your set point is 71, or I think it is, and then I measure the temperature in your house and it's 90, what I know is about to happen. This isn't rhetorical. Temperature's going to go down.
Or the ASIC.
Exactly, right. So if we think that the temperature is the thing that matters and the set point is 71, and we can measure an error signal around that set point, we should be able to predict the future behavior of that system. And so that's the thing that nobody's done yet in the field of sleep. We've got a lot of measures that seem to go up and down during sleep and wake, right? But nobody can predict the future behavior of the system. And so that's exactly what we set out to do. So this is all the work of Ifan, who I'll show you a picture of later. But here's an example of a few hours of animal going between sleep and wake, sleep and wake, sleep and wake. And so Ifan sort of algorithmically searches across thousands and thousands and thousands of hours of recording, and he finds what he calls wake dense and sleep dense windows. So this is 1 hour that is more than 2/3 awake, or 1 hour that is more than 2/3 asleep. And then he extracts the dynamics of the system from the preceding 2 hours. So in that prior 2 hours, he looks at our measure, DCC. He looks at how fast neurons are firing, how variable they are, some other things that people have posited and predicted in the past. He says, well, we're going to pull out all of those things. And instead of saying, people know this, neurons tend to fire faster when you're awake than when you're asleep, right? But does that actually give you predictive value? Does it accumulate and predict the future behavior of the system? So that's what we're really trying to know here. So we can then say, once we know these dynamics, do we have any information about the future behavior of the animal? So it turns out that if you have DCC in the 2 hours prior, so bin A is the prior 2 hours, and bin B is your sleep wake dense period. DCC preceding a wake dense period is lower than a sleep dense period. So it's kind of consistent with what we might predict, right? So for an animal, so let me just unpack that for a moment. So we would think that for an animal to be awake for a while, its DCC should probably be low, because if that kind of distance from criticality is going to drive it towards sleep, a higher DCC is going to bias you towards sleep. So this fits with that. So we can actually flip that and do that the other way. If we take the DCC in A and then say, what's the probability of seeing a sleep or awakeness in B, we see it's a log normal, like very, very, very clear relationship there, right? So if we know that DCC is lower in the first window, we can make that prediction about the second window. And then finally, and the key point here, this is not true if we use these other measures. This is not true of anything else that we've tried looking at in the brain. So if we pause for a moment, let me just sort of summarize this. We have a theory predicted set point that you can come into ahead of time and say, we know this is a homeostatic target in the brain, and it directly explains function. It directly explains computational capacity, which is ultimately cognitive and behavioral capacity. And then we say we expect that to change around this set point in sleep and wake. And so far, it looks like that's true. So then we can build a logistic regression, which is basically a simple classifier that just operates on one variable, and ask, we shuffle data, we'd expect to be ever so slightly above 50%. Chances, animals, I think they're awake slightly more than they're asleep at this age. And the key thing to note is that none of these measures, so this is the firing rate of neurons, for example, this delta wave is the thing that people have looked at for a long time trying to confirm Borberly's model. None of these things give you any insight into whether or not you're about to hit a sleep or awakeness period. The only things that seem to matter are Zeitgeber times, that's the time of day, right? So now we're getting at that process C. The prior behavioral history then, just knowing that it was awake before gives you some predictive value. But then the strongest thing that we have is DCC. So we can take the same data set and go a little bit further with it. Because it's unlikely that these variables are sort of operating independently. So the base model that we talked about, process C and process S, is the combination of the time of day and the behavioral history of the animal, right? So I'm gonna make a sort of a two-component variable called base, where we're combining time and prior history. And then what we can look at is, here's the shuffle data, and simply knowing that base model, this is the heuristic model as close as we can get to it. You do a lot better. I mean, that's at 65%. So then we can start adding things to that to say, do we get past that base model? And none of these variables matter. So we can add firing rate, variance, that delta wave, we can add all of that on top of base model. It does not improve the performance of the thing at all. But then when we add DCC to it, we get a significant improvement. And then if we just take all of the variables all together, we don't outperform DCC. So what this tells me is that DCC is giving you information that is not redundant with the behavioral history of the animal. And DCC is the only thing that we can find so far that lets you predict the future behavior of the system. We can then take that final model here and decompose it and look at what is it actually trying to learn from. And you can see that the weights is like a sort of machine learning algorithm, right? You can say, well, where are you putting your weight? What are you actually paying attention to, right? And you can see that it's exactly what you predict. Time of day, behavioral history, but most importantly, the system's learning from DCC. So this is kind of, again, consistent with this hypothesis that this should be the fundamental set point, that the brain is not attending to, I would argue, some protein level going up, down, or a hypnogenic substance. It's attending to its computational capacity. And there's something about waking, and I can tell you why I think that's happening, but there's something about waking and experience that progressively pushes networks away from that set point. And that has to be true. And I can explain it later if you guys have it in you. But sleep is bringing that back. So we can then make 3 further predictions about this. So first, if we're right, so right now I just told you we can predict the behavior based on a single measurement, right? Like if I know DCC in that 2-hour bin, we have some predictive value. But we can go further and say changes in this error signal should be progressive. So as you stay up later and later and later and later at night, I would predict, oh, I'm wearing a microphone. I would predict that your DCC should creep up with that, right? Similarly, the longer your sleep, the more DCC should come down. Second, the error signal should not just be a clock. And this is intuitively obvious if you think about it for a moment, because being awake for 12 hours on a Saturday, it's rainy, there's not much to do, you're watching some movies, that is not nearly as taxing. I don't sleep nearly as hard after that day as a day where I have to teach my first class of the semester, give a talk, write a grant, all this stuff. Then I feel like I could sleep for 14 hours, right? There's something about the content of experience that matters. It's the burden of experience that is variable. So we need to be able to dissociate just a timekeeping mechanism from the content of the experience. And then that's kind of recapitulated here, is that you can also say that as that the context of that waking experience should have some value in this. So here's how we run this experiment. We take 4-hour windows where an animal is awake or asleep. And this is very tricky to do, again, because we're trying to use free behavior. And then instead of looking at it in terms of 4 hours, you could divide it into the percentage of time. So that's 0 to 100% of those 4 hours. And then what we do is we concatenate the wake time and we concatenate the sleep time. And then we measure DCC in here. DCC in here. So that way those sleep and wake bins are not contaminating each other, but we're going to have variable amounts of time that were spent in each of those. And so what we predict, that there should be some relationship between the amount of time in that 4-hour bin that was spent within the state and the DCC within that state, right? So again, imagine if you had 3.5 hours of that as awake, I would predict that even though it's going to be really noisy, you would expect to see a meaningful increase in DCC because of that amount of wake. So we should be able to regress this now. So here's what we get. Here's DCC during waking as a function of time within that state. And we have a significant relationship there. And the key point is that that's in the light, because we don't see that in the dark. And this is in the primary visual cortex. So what I think is happening is that when you have external input impinging on a system, it changes that system. The fact that you can remember what I just said, the fact that you know where your car is, what you had for lunch, the quarrel you had with your partner this morning, all of that revolves around changing the weights and the connections and the proteins in that system. You are fundamentally changing the system when you use it. And so when you change the visual system by using it for visually guided behavior, you push the system away from criticality, and the same amount of behavior, the same amount of locomotion, the same amount of eating and grooming the mouse is doing in a completely dark environment does not have that predictive effect on the system. And if we look at active waking, so we know that when that mouse that I showed you chasing the cockroach, we know that's a visually guided behavior. So if we look at the effect of active waking, it gets even stronger. So when the mouse is specifically restricted, we restrict our analyses specifically to periods where the animal is using vision to guide locomotion, we have a stronger relationship, and that falls apart more in the dark. And if we look at quiet waking, so this is where we call an animal they spend a lot of time doing this. They just sit. They're mice. They don't. It's a mouse. We see that this falls apart, right? So you're not it's not using visual information here. Interestingly, exactly as we would predict, whether it's in light or dark, sleep brings DCC down. Okay, to be thorough, we check everything. Because again, we kind of collected everything. So we can apply the same exact analysis to firing rate, to variance, to all of the things that other people have sort of suggested in the past, and we see no relationships. So here's an example of the sort of variability of neural signaling in the brain. We don't see that same pattern here. And here's firing rate. We don't see that pattern with firing rate. So the problem that I run into, and Eric Herzog does this to me all the time, he's like, can you show that it's causal? It's a cute correlation that you have. And I hate the idea of causality in complex systems, especially dynamical systems. Like I'm not sure what that even means. Right. So think about for a moment like a pendulum is swinging. Like what's causal there? Is it the weight? Is it the cord? Is it the pivot? I mean, all of it, like everything, right? I think this is more like astrophysics. You need to observe a very, very complex system and make specific predictions about how it should behave if you've figured it out. And if those predictions bear out, then you've got it. I mean, think about relativity, sort of everything like gravitational lensing, all of that operates on that principle. We can't interfere with, other galaxies to sort of test the causality of black holes, but we still believe they're there. Okay, sorry, that was my little aside for you. But that being said, rather than dumping a bunch of drugs into an animal's brain, rather than sleep, so the field standard is sleep deprivation. Have you guys ever been sleep deprived? It's like 4 of you. This is really, LEC seems like a good place to live. Okay, sleep deprivation sucks. It is awful. There's no way you're going to convince me that. Let me put it this way. Me being awake under normal conditions is not the same thing physiologically as me being awake after 26 hours of sleep deprivation. You're not studying the same thing. You are looking at a system in different conditions. So that being said, it's the ruler that everybody uses, like, oh, you don't sleep deprivation. So we have to do something. So instead of keeping an animal awake for 6 hours, which to a baby rat would be catastrophic, we bought Ifan night vision goggles and a lot of Red Bull. And right, yeah, right. So what Ifan did, here's an example of 20 hours of Ifan's life playing with baby rats. So every time, let's start here actually. Here's a cumulative distribution of the duration of waking epochs. And in orange is the natural durations that we see, right? So sometimes left to their own devices, animals will be awake for 150 minutes. So what Ifan decided to do is every other time the animal is awake, he just keeps it up for 90 minutes. So we're going well outside of the mean, but we're not going past what these guys can do anyway. And then he lets the animal get a recovery sleep, have a normal waking that's unperturbed, it sleeps again, and then the next time it's awake, he puts new toys in its cage or he brushes some food around. So it's not hard to keep them awake for this, but the idea is to say, can we show that if we impose waking on these animals, do we get the effect that we assume we will get? Right? I would say that's the sort of the closest thing we can do to sort of causal sleep deprivation manipulations without really making me upset. So again, here's DCC across 190. This is an example of one animal, and Ifan here in the light is not wearing dark vision goggles, and then here he is overnight wearing night vision goggles. So you can see DCC goes up, and then the animal has a recovery sleep here. It comes down, it goes up, and then it comes down during sleep, up, down, up, down. It's exactly what you would expect, right? So we can say this isn't just some weird, bizarre timing correlation, but when we actually drive waking in the animal, so he divides each of these into 3 periods. There's the start, the middle, and the end, and then the recovery. And you see consistently, both light and dark, that the system increases its ECC and then that then recovers. And the effect is a bit stronger than in the light than in the dark. Yeah.
Why, is it just because of the time points that it kind of starts, DCC starts sort of returning toward the baseline before the sleep starts?
You mean here?
Yeah.
Yeah, it's just the binning, right? Like he has to, he's just taking it in sort of 30-minute segments. So this is not a continuous time series. He's just measured DCC here, and then he's measured it here. So the point at which it switches. So this is an interesting and sort of more methodological concern, but to collect, so remember the blue swirling dots, you need to measure a lot of those events passing your electrodes before you can really estimate the system, right? And so the problem then is that you need a good amount of time before you can get a solid estimate of those power loss. And so EFON can't do a 2-minute rolling window. He's got to take these kind of 30-minute blocks, and that's already stretching it. Like usually we use 1 to 4 hours. So that's exactly what you're noticing. Okay, so, oh, great. I've got 8 minutes and I wanted to get to something totally different or totally new in the lab. So I did. And again, it's not ecology. So, okay. So this raises an interesting question that we've been pondering for a while, which is, how do you know that criticality is the set point? How do we know that, so what if it varies with this and it has these predicted values? How do you know that it actually has anything to do with animals' ability to think, an animal's ability to perceive, to detect, to sense, to generate complex behaviors? How do we know? We don't. The best thing we have is modeling, the best thing we have is theory. We can show that the brain is critical, but to actually, I guess I don't want to undercut my work. I think that the sleep thing is pretty strong, right? I think that seems to resonate with me. But how do we know that it's the set point that relates to the animal's behavior? And so I said we can kind of invert this question and say, well, what if we take an animal that's broken, right? Because if criticality is the set point that explains normal behavior, or that is a predicate or the necessity for normal behavior, then when you have an animal that cannot generate normal behavior, if you were to see that it's critical, then you're dead in the water. You know, your theory just went out the window. So to do this, that guy, James, did a very hard series of experiments where he implanted microelectrode arrays into the hippocampus of mice. And he recorded from these for 27,000 hours. So he generated a petabyte of data. He is part of the reason that we are renegotiating our contract with Box. Because 6 of us have maxed out. It turns out it is not an unlimited storage contract. It is very limited. There's a hard line, and we have maxed out 6 lab members' accounts on Box. And that's the guy in the yellow shirt who's responsible for that. So that's Kieran. Okay, so here's a timeline of James's recordings. He starts at about 50 days old in the mouse. Each of these little bubbles represents a mouse's life. And then he records about postnatal day 550. That is a very old mouse. And the key thing is that the gray mice are wild type, but the yellow have a human tauopathy transgene. Tau is one of the key molecules in a bunch of neurodegenerative disorders, including Alzheimer's. And so what happens here is that these animals start out looking just fine. And then as they go out throughout life, the yellow mice develop severe neurodegenerative disease. They get big pieces of the brain start to collapse. There's a bunch of misfolded proteins. Neurons start to die. They have major behavioral problems, and then they die. You'll notice that we can't get any gold animals out here at this point in life. And so our prediction is the following, that if we've got this right, that you should be able to predict the genotype of an animal and the age of that animal by looking at this setup, right? So you don't need to biopsy a brain. You don't need to pull out a bunch of molecules from a bloodstream. You should be able to look at the operating system of the brain, because if there's a failure in cognition, a persistent failure in cognition, there must be persistent failure in whatever the set point is that determines computational capacity. So that's what James set up to do. So here's just, we're going to skip this. This is showing you that he can record. Well, we're not going to, I'm not skipping it, I'm doing it. He's showing you that, because a lot of people won't believe this, that in very, very old sick animals, he can record the activity of individual neurons. So nobody else in the world does this. This is James through and through. Most people record for a few hours at a time, and they do it in sort of an animal that's maybe 60 to 70 days old. So the fact that James can do this here is very, very, very much out in front of the field. Oh, and I'll also point out the reason that he kills the animals intermittently like this is so that he can then look at the underlying molecular biology. He can map the dynamics back onto the decay inside of the brain, right? So we have behavior, we have molecular biology, and we have dynamics. I should say James has those things. So the key thing to start with is set points that we know exist in the brain, things like firing rate. So you might expect tau builds up inside of neurons. It's an intracellular, it's awful. It looks like, it literally looks like, I have a cat. There are 3 cats in the LEC. You got 4. Okay. So have you ever seen a hairball? You ever look in your shower drain? That's what tau looks like under a microscope inside of neurons. It's disgusting. So it builds up inside of neurons and it kills neurons. So you might expect that a neuron level set point is the thing that's likely susceptible to damage by tau. And so the rate at which neurons fire, the number of spikes per second, is a crucial set point in the brain. What James sees is that here's a young wild type animal, here's an old neurodegeneration animal. He has 2 different cell types that he can track across 12-hour light, dark, light, dark, light, dark. It's 2 weeks for each one. It doesn't, but things are stable in both animal, and it doesn't look like they're any different. Then he could break that down and look at the distributions across young, middle, and old, both genotypes across life. This is not changing. So this is a stunning negative result on its own to suggest that these homeostatic set points, this is a different thermostat, are completely robust to disease, which is shocking. But then in the same exact data sets, when James turns to criticality, so again, here we can get all the spikes, we can look at those swirling blue patterns. Do you remember I told you, we look at the alignment of the predicted line and the data? You guys with me? You remember that? We did that together? It meant a lot to me. Here's a young wild type animal. Here's a first power law, here's their second power law. The black is the predicted line, the pink are James's real data. Does the fit, he sees that DCC of less than 0.2. So then if he looks at an old TE4 animal, that animal is fucked. Okay. And so I'll say this as I wrap up. This changes just before the onset of the earliest symptoms. We can predict the underlying molecular biology, we can predict the underlying anatomical biology. He can predict an animal's genotype with 90% accuracy, or 80% accuracy with the updated models. And this is the only thing that we know of that's changing in the brain, which is crazy. Just think for a moment, I'll close on this. A disease isn't a protein. It's a protein. A protein is a protein, right? A disease is, by very definition, the failure of computation in your brain. Because if you didn't have a failure of computation, you just say, well, some people have tau in their brain, no big deal, right? It's the effect of that protein that is the disease. And if you ask Alzheimer's experts, et cetera, well, what's different, consistent about brain activity in somebody with Alzheimer's? They say, we have no idea. Because when you look at traditional metrics, variance, firing rate, synchrony, those things are robust. They are not changing. This is. So with that, I should thank a few people. So I showed you data at the beginning. Jacob and John got a lot. How many cockroaches have you guys gone through? We are the world's #1 consumer right now of Tajikistani regular cockroaches. I'm not kidding. It's disgusting. Thousands of cockroaches. Yvonne did all of the sleep DCC criticality work, and James is the one who's responsible for babysitting a lot of animals with progressively worse neurodegeneration. So with that, it's 5 o'clock. I'm out of time, and there's beer across the hall. I think I just saw two questions. Yeah.
Okay.
Joe, that's all you.
Have you handed this,
What?
What is the question right here? Round of a lot of hands. Let's make sure it's on.
Yeah, it was on, buddy. We established that.
Okay.
I was wondering how to reconcile your like sleep fraction plot with there's some old classic working like circadian clocks in mice, or like when they measure when they like run on a wheel and in the dark, and it's like this very nice, like they always wake up at this time and start running. Is that a different species, or mice versus rats or something? Like
Yeah, that's a really good point. So just for everybody else, the question is, when you look at the original literature, people can measure kind of the circadian distribution of behavior in mice and rats by putting a running wheel into the cage and then just measuring when that goes. And they will run 10 kilometers a night. That's a lot for a mouse. It's a lot for me. So it turns out that when you put that wheel in there, it changes their behavior. Right, so it's kind of like if you leave a 14-year-old near an Xbox, they will stay up a lot later, right? So it's very much so, like if you put the mouse near that wheel, it's like, let's do this. Okay? That, and we are working in the sleep study in rats that are roughly 30 days old. And so they don't have quite as consolidated a cycle. So the key point is that even in an adult rodent, sleep is much, much more fragmented than you might have been led to believe. But that effect is heightened a bit earlier in life. And the wheel is like crack. So, they will run until they die. I have a border collie. It's very reminiscent of my border collie. It's just like fixation.
I have two.
Yeah, okay, so you know what I'm talking about. Yeah. Yeah. If you ever been in a lab where they have those, it's so annoying because the lights go off, they wake up, they're like wheel time, and you hear beep, 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 like in the background. You're like, I'm gonna get rid of this wheel. Shifting fields. Okay, sorry.
Give me one more question I'll give out, and then you can ask Keith more questions just across the hallway. We have drinks and refreshments and some food. Anyone else? Maybe we should call it 5, one hand.
Really quick question.
Yeah,
Sure, I guess.
I'll just repeat it.
Yeah, so I'm just curious, on the last slide, you showed the difference between the young, healthy
Mhm.
animals and the old, sick animals. Is there some sort of baseline for how that DCC value changes as the animals age? And do you think that that's something that also might be repeated in humans?
Yeah, so I'll start with the second part first. I suspect that this will be true in humans. Like, I think that there's a reason that we use model species. I think, evolution matters and things are conserved, and people have seen signatures of criticality in humans, so I suspect that this will be true. The cool thing that James has, is very unique on the data set, is that lifetime recording, right? That sort of 27 or 30,000 hours of recording. So what he sees is that just at sort of the first detectable moments where misfolded tau would be detectable. If you were to do like a whole brain homogenetic, kill the animal, take out the brain, and like do really hardcore, high resolution molecular biology, at that time, we start to see that variability increases. So it seems like the set point is still there, right? Like the thermostat's still set to 71, but whatever the mechanisms are that normally restore that and maintain that start to fail. So it starts to have bigger deviations and more frequent deviations from 71. And that is totally detectable. It's sort of like very, very clear time course where the system just takes off and oh, that animal's sick.
But it's not related to like a healthy mouse at the end of its life would have a different measure of BCC than a young mouse.
That's actually a really good question, and I guess we can go across the hall. So one of the big questions in neurodegenerative disease that I think has been answered is the following. Is neurodegenerative disease just an advanced aging process? As in, if you could live to 200, you would get this thing. And I think the answer is pretty clearly no, it's not that. It is truly a unique phenomenon. In our data, the old animals actually look like they're getting closer and closer and closer to criticality, right? So almost the exact opposite. And I can speculate on why that might be, but yeah. So an old wild type animal does not begin to recapitulate the disease effects.
Great, well, let's end it there. Let's give Keith a round of applause.